Good afternoon, friends. Now, in the last some lectures, we have learned about the series of UV visible spectroscopy chapter. Here, we will again continue the UV visible spectroscopy chapter, but the process, or you can say the theory involving this in this video are different than the remaining one, because in the last some few lectures we have learned about the instrumentation, basic instrumentation involved in the colorimeter and spectrophotometer or any type of photometry. But in this technique, in this video we will learn about different one. Here we will learn general procedure for colorimetric estimation. So what are the general procedures are involved in colorimetric estimation? So what should be the care to be taken when the colorimetric estimations are to be handled? In a part by the instrument to be used and nature of the substance to be determined. Now here, which type of instrument is used in the colorimetry and which type of the substance has to be used in colorimetry or any type of photometry, or can say the colorimetric estimation. Especially here, colorimetric estimation are mentioned. So the type of the instrument and the type of the substance or nature of the substance govern the colorimetric estimation or depends on the colorimetric estimation. So colorimetric estimation basically depends upon the first point, that is the instrumentation to be used and nature of the substance to be determined. The second point, there are some general principles that are uniquely and universally applicable and are also useful in spectroscopic determinations. Now, if you look, there are some basic principles which are common in any type of colorimetric estimation. For example, colored substance that should be used. Now, in colorimetric estimation, colored substance has to be used. If the substance is not colored or color solution is not colored, then that analyte has to be bring into a color form. So it is needful that colored substance or color solution must be used in colorimetric estimation. So that is a universally acceptable principle. So here the second point is that there are some general principles that are uniquely applicable and are also useful in spectroscopic determination. Now the third point, operating handbook supplied by manufacturer gives the specific technique to follow in such an instrument. So always when the any type of colorimeter instrument we will purchase, at that time we get the handbook, that is operating handbook, how that colorimeter is used, and that operating handbook has to be supplied by the manufacturer. So with the help of that handbook, we will get the information about the path or how to handle the instrument, such a type of instrument. The fourth point is that it is clearly to see whether colorimeter or spectrophotometer is functioning correctly or ensure no adjustment or replacement is required. So the care should be taken that colorimeter or spectrophotometer should be handled very carefully. If some part of the colorimeter or can say spectrophotometer is deliberately or can say is manually replaceable or adjustable, then that we have to ensure that proper adjustment or replacement has to be done or not. So here fourth point is very most important. If any part is harmful or can say it will be get disturbed by the use of some long time, then at that time that functioning or can say proper proper functioning is also needful, and proper adjustment or replaceable of such a type of manual or such a type of such a type of parts in the instruments has to be replaceable, or can say it is required conditions. In absence of automated facility, the following four things should be examined. So following four things, which are the four things that should be examined when the proper requirements has to be done with the adjustment and replacement? Then these are the four things, that is illumination, wavelength scale, absorption scale, and cells. Now illumination means source, that is a source of light. In absence of automated facility, now some of the colorimeter or spectrophotometer, if you take into account or if you take into consideration, then at that time there is a manual automated system has to be performed to take the reading, or we can say to take the output of the colorimeter or spectrophotometer. But if automated system is absent, then in such a condition, colorimeter or can say spectrophotometer, then there should be necessary conditions are there. These necessary conditions are nothing but the proper examinations of 4 parts, and that proper examination of 4 parts are nothing but the source of light, that is illumination. Any emission of lamp decreases with age. When the time of maximum utilization has passed, a record of each lamp's burning time should be kept. Now, when the source of light is new, there is no problem. But if the source of light becomes old, at that time the problems get encountered, because any emission of lamp decreases with age. So when we purchase and when some time has to be completed, then that proper any type of, we can say, any type of luggage or any type of source of light or any type of lamp, that its life decreases with time. And so care should be taken that when the time of maximum utilization has passed, when we use maximum utilization of that particular lamp, at that time we should replace that source of light, because with the increase in age, with the increase in time, its intensity, its usefulness decreases. A record of each lamp's burning time should be kept. So when the system is started for colorimeter, at that time burning lamp should be open, or you can say it will be on. At that time, how much time it will be started in for a particular time, that time should be recorded. So this is first point, that is the illumination or source of light. Wavelength scale, second point. So wavelength scale should be properly calibrated. Wavelength scale should be properly calibrated. For a better result, the filter that come with instrument will be used. Now, for the wavelength scale, no doubt, the wavelength scale is very important, and the wavelength scale is also determined from the value of lambda max for the any type of filtered. So the filter that come with the instrument will also used. When we purchase the instrument, at that time filters are also we will get, or manufacturer provides the filters. So for better results, the filter that come with the instrument should be used for the calibration of wavelength scale. The third point in this regard, absorption scale. This can be verified by using one or more carefully prepared standard solution, such as potassium dichromate in either acid or alkali solution and potassium nitrate solutions. Now, absorption scale is also important like wavelength scale, but the absorption scale can be verified by using one or more carefully prepared standard solution. Now, what is a standard solution? Standard solution is nothing but it is a substance, or can say it is a solution whose concentration we know, whose concentration is known. That is called as a standard solution, and that can be we can say such as if we prepare a potassium dichromate in either acid or alkali solution and potassium nitrate solutions. At that time, with the help of these two solutions, absorption scale that can be verified. Now the fourth one is that this cells. Here cell is there. Now two selected cells displays the same transmission within the narrow limits under identical conditions. Now if you take the two cells, then there is a correction that should be done, and with the help of this correction, two selected cells display the same transmission within the narrow limits under identical conditions. Cells can be accepted in use when difference between them have absorbance is less than 0.02. When we compare the cells, if we take the absorbance of that two cells and we have to use that two cells, then in that two solutions, or you can say in that two cells have the comparative absorbance value, that is 0.02. So these are the 4 points. These are the 4 points that should be useful when we examine the scale or different measurements for the colorimeter. So this is all about the first part. Now the second part of this video is nothing but the choice of solvent. So solvent is useful to dissolve the substance, and that prepared solution is useful to measure the intensity of transmitted light. And this measured intensity of transmitted light is done with the help of detector. So detector detects all transmitted radiations, and we know that photomultiplier tube, photovoltaic cell, and silicon photodiode. These are the 4 detectors employed in colorimeter and spectrophotometer. Now here are the identical solvent is nothing but there are some conditions are given. A suitable solvent used in colorimetry and spectrophotometry are as under. First, it should not itself absorb radiations in the region under investigation. So if a region is under investigation is there, then at that time that solvent should not absorb the radiations. The second is that it should be less polar, so that minimum interactions with solute molecules. So solvent should be less polar. Second condition. The third condition is that it should be good solvent for substance under determination. So whatever the substance we have to determine, at that time it is useful for that. The solvent should be useful for that substance. The solvent should be good for that substance. So these are the 3 conditions. First is that it should not itself absorb the radiations. त्याने कोणत्याही प्रकारचे रेडिएशन्स ऍब्सॉर्ब करायला नकोत. सॉल्व्हंटने कोणत्याही प्रकारचे रेडिएशन ऍब्सॉर्ब करायला नकोत. दुसरा असा आहे की सॉल्व्हंट हा पोलर नसावा, तो कमी पोलर असावा. तिसरा असा आहे की इट शुड बी गुड सॉल्व्हंट, तो चांगल्यापैकी चांगला सॉल्व्हंट असावा, तो त्या सबस्टन्ससाठी चांगला सॉल्व्हंट असावा. फॉर इनऑर्गॅनिक कंपाउंड्स वॉटर कंप्लीट्स द रिक्वायरमेंट्स, while non-polar compounds, hydrocarbon solvents like phenol in cyclohexanes are used. So water is useful for the inorganic compounds, no doubt, but for organic compounds hydrocarbon solvents are selected. For example is there phenol in cyclohexane. So this is all about this you can say choice of solvent and measurements, whatever the measurements has to be determined in colorimetric estimation, in which way measurements are depends. So there are the 4 factors: cells, absorption scale, wavelength scale, and illumination or source of light. So this is all about this video. With this content I conclude this lecture. Thank you very much for patience listening.